Hello, and thank you for joining the Sonodynamic Therapy webinar hosted by the Focused Ultrasound Foundation and the National Brain Tumor Society. My name is Lauren Polovich, and I am the Associate Chief Medical Officer at the Foundation. I also direct our Sonodynamic Therapy program. For those of you not familiar with the Focused Ultrasound Foundation, our mission is to catalyze the development and clinical adoption of focused ultrasound technology. We operate with a sense of urgency that the time is now to get this revolutionary technology to patients. We accomplish our mission through the creation of knowledge via supporting preclinical and clinical research, advocacy and education events, cultivation of the next generation through internships and fellowships, and bridging gaps with focused ultrasound startup companies through our Focused FUSF Partners branch. Since focused ultrasound has the potential to impact patients suffering from a wide variety of diseases and conditions, the Foundation has created specific programs and initiatives to hone our resources. The Sonodynamic Therapy program is one such program. We support preclinical and clinical research in SDT. We recently held a roundtable discussion to answer burning questions on how to most successfully proceed with the sonodynamic therapy field. And we've included SDT in recent workshops related to brain tumors. We are very happy to have the National Brain Tumor Society as a co-host for this webinar. And I'm going to hand over the mic to Kirk Tanner, the Chief Scientific Officer at NBTS, to give a brief overview of this very important organization. Thank you, Kirk, for being here.
No, thank you so much, Lauren. It's really been great working with you, Susie, and the entire Focused Ultrasound Foundation team. This is a really important topic, and I'm very much looking forward to the presentations from our speakers today and discussion around sonodynamic therapy. So welcome, everybody. Thank you for joining the webinar. As Lauren mentioned, my name is Kirk Tanner, and I'm the Chief Scientific Officer of the National Brain Tumor Society, NBTS. NBTS was created more than 30 years ago by families who felt helpless after losing their sons to brain tumors. In the midst of their pain, they asked themselves really the same question: what can we do to help fight back against brain tumors? To that end, NBTS and its legacy organizations were formed. NBTS has a long and accomplished history of funding groundbreaking neuro-oncology research and building the modern brain tumor movement. In 2023, nearly 1 million people in the US are living with a brain tumor, and they need better treatments and improved quality of life, and ultimately cures. NBTS is here to support, convene, and advocate through our 3 programmatic strategies: defeat, connect, and change. Defeating brain tumors requires focused efforts and comprehensive strategies. Our unified and multifaceted approach enables us to create change across policy, research, healthcare systems, and drug development pipelines at all levels, tackling roadblocks to find a cure. We're here to kind of reach those next breakthroughs and defeat brain tumors once and for all, to connect patients to each other and, more importantly, to all the stakeholders, and to champion for more funding from federal government to fight brain tumors so that we can all create real and lasting change. NBTS focuses on groundbreaking research initiatives with the potential to translate promising science from the lab into treatments. We see how a non-invasive treatment option like sonodynamic therapy could be a game changer for high-grade glioma patients to not only improve survival, but quality of life. So in today's webinar, we'll learn more about this therapy from experts and also the latest updates on where the research and clinical trials currently stand. So with that, I'll turn it back over to Lauren. Lauren?
Thank you, Dr. Tanner. Now, Dr. Suzanne LeBlang, the Director of Clinical Relationships at the Foundation, will give a brief overview of the state of clinical trials in sonodynamic therapy before diving into the presentations by Drs. Agarwal and Prada. Dr. Prada is the Director of Neurosurgery at the IRCCS Foundation Carlo Besta Neurological Institute in Milan, Italy. He has led pioneering work in SDT with both 5-ALA and fluorescein, which you'll hear about today. Dr. Vijay Agarwal is the neurosurgeon and founder and CEO of Alpheus Medical, a company that is harnessing the power of SDT to treat glioblastoma. Following their presentations, Doctors Prada, Agarwal, and LeBlang will join for a live moderated question and answer session.
Hi, my name is Suzanne LeBlang, and I'm a neuroradiologist and the Director of Clinical Relationships at the Focused Ultrasound Foundation. I'd like to share with you the current clinical landscape for sonodynamic therapy. I'm encouraged to report that there are 5 clinical trials currently enrolling patients for sonodynamic therapy, and 1 about to begin at UVA. Sonodynamic therapy is a non-invasive drug-device strategy that involves the sensitization of target tissues, such as brain tumors, with a non-toxic chemical agent, commonly 5-ALA, and subsequent exposure of the sensitized tissue to low-intensity focused ultrasound, or LIFU. Both aspects, the sensitizing agent and LIFU, are harmless until combined, when cytotoxic events such as cell death can occur. The 3 companies within this space are Sonodynamic, Alpius, and Navifus, and they each use a different focused ultrasound machine. Now, the first row is a study performed by Dr. Nadir Sinai at the Ivy Brain Tumor Center. He conducted a controlled first-in-human phase 0-1 clinical trial to evaluate the safety and potential for rapid biological effects of sonodynamic therapy using intravenously administered 5-ALA in combination with the Exablate Type 2 device by Insightec on patients with recurrent glioblastoma. 10 patients participated, treated across 3 escalating energy doses. Half of each tumor was treated with focused ultrasound, the other half remained as a control. 4 days after sonodynamic therapy, the complete tumor was removed by surgical resection, and each of the treated and untreated tumor halves were studied and compared for sonodynamic therapy effects. There were no serious adverse effects related to the drug or device. There was 1 serious adverse event of short-lived hypotension in a patient post-IV infusion, which was unrelated to treatment. Energy doses up to 800 joules were safely administered, and preliminary results showed statistically significant elevation in biomarkers for apoptosis and lipoperoxidation in treated tumor compared to controlled. Full results are pending publication. On the second row, using a similar combination of IV 5-ALA and the Insightec FUSF machine, Sonodynamic's ongoing phase 1-2 clinical trial for recurrent GBM is recruiting at multiple sites. On the fourth row, Sonodynamic is the first company to perform sonodynamic therapy on DIPG, a pediatric brain tumor mainly affecting children between 2 and 9. This is a devastating diagnosis as current treatments aren't effective, with median survival of 9 to 12 months post-diagnosis. This ongoing phase 1-2 clinical trial in DIPG involves an initial dose escalation in 9 children and then a dose expansion phase in up to 18 more participants. Monthly sonodynamic therapy treatments are given. To date, 5 DIPG patients have been treated and it has been well tolerated, with no major drug or device-related adverse events. Importantly, patients also showed a maintained quality of life and consistent performance status scores. The first publication related to sonodynamic therapy in a clinical trial was reported earlier this year in the Journal of Neuro-Oncology. These authors from Children's National Hospital reported the first patient with DIPG who underwent sonodynamic therapy to 50% of the tumor within the right side of the pons. The patient tolerated the procedure well, and post-procedure MRI scan demonstrated no adverse changes on T2 images compared to the pre-treatment scan. In row 3, there is a novel trial in Italy using the oral formulation of 5-ALA along with the machine from Insightec for de novo glioblastomas. We will be hearing from the neurosurgeon Dr. Prada about this trial. In addition to the machine by Insightec used for the first 4 clinical trials, another company performing sonodynamic therapy in clinical trials is Alpius. We will hear from Dr. Agarwal, a leading neurosurgeon, about why his approach with sonodynamic therapy is different than the studies I just mentioned. Finally, Navifus is anticipated to start their clinical trial at UVA in the near future with patients that have recurrent GBMs. Their preclinical studies have showed quite promising results. Interestingly, although the 3 machines used in clinical sonodynamic therapy trials all use focused ultrasound to activate a sonosensitizer like 5-ALA, the method of delivering the focused ultrasound is quite different. The first machine, manufactured by Insightec, uses MRI guidance, so there is a helmet around the patient's head, and while inside the MRI machine, the focused ultrasound is delivered. The company Sonodynamic uses the drug-device combination with an IV formulation of 5-ALA, while Dr. Prada in Italy is using an oral form of 5-ALA with this device. The NaviFUS device uses neuronavigation. That is, a previously performed MRI scan is loaded into the computer, and utilizing optical guidance, a focused ultrasound transducer is guided to the proper position over the target region using that MRI scan. Since this is not performed in an MRI scanner, it can be performed in an outpatient office setting. Finally, Alpheus delivers the focused ultrasound beam without any imaging guidance and does not even require a prior MRI to be performed. This can also be performed in an outpatient office type of setting. I hope this information serves as an overview, and now let's hear from the other speakers.
Hello, everybody. So I will talk a little bit about the concept of sonodynamic therapy and its clinical application in glioma treatment. As most of you will probably know, glioma, especially high-grade glioma, they account for 80% of all malignant brain tumors. And despite a combined treatment that entails surgery, chemotherapy, and radiotherapy, their prognosis is really dismal. So one of the mainstay of glioma treatment is often surgical resection, and it is important to remove the bulk of the tumor, sparing the neurological functions. And to this end, there are many imaging tools. Image guidance is very important. A very interesting optical modality is fluorescence-guided surgery. And one of the compounds used for fluorescence-guided surgery is 5-aminolevulinic acid, 5-ALA, which is basically a precursor of protoporphyrin 9, and it is selectively metabolized and accumulated within the tumoral cells. And when highlighted with a certain wavelength, it becomes bright orange, as you can see in the picture, showing the tumoral tissue. And as a matter of fact, there have been many studies. Mainly this technique was popularized by Dr. Stummer, that shows its efficacy to show the bulk of the tumor. As I mentioned, 5-ALA is a prodrug, which is naturally produced in the body, and it is an intermediate in the synthesis of heme. And because of enzymatic failure within tumor tissue, there is a selective uptake of 5-ALA, which is transformed into protoporphyrin 9, which is actually the fluorescent compound. There are two formulations of 5-ALA. The one that was approved in Europe in 2007 is the oral formulation of 5-ALA, which goes under the name of Gleolan, and it has been approved in 2017. And for sonodynamic therapy, there is another formulation which is IV administered, which is called Sonodynamic. There are, of course, other fluorescent agents that can be used for fluorescence-guided surgery, such as fluorescent, which works in a totally different way because it extravasates across the open BBB within the tumor. It can also accumulate in the brain and in the other tissues, so it's less specific than 5-ALA. It is interesting because it can be visualized in other type of tumor other than gliomas. Most of the studies for the use of 5-ALA, which we know selectively accumulates in the tumor, in order to exploit this feature of the sensitizer of being selectively localized within the target zone, is to exploit this drug for therapeutic purpose. So photodynamic therapy is a very interesting concept, which is a precursor to sonodynamic therapy. And the idea is basically to activate the drug, which is already selected by itself, with a form of energy, in this case, with light. And doing so, we can change, the idea is to change the state, the state of activation of the substance, generating reactive oxygen species, which in turn they induce cytotoxic reaction, necrosis, and apoptosis. One of the downside of photodynamic therapy is the light penetration is limited, usually to 5 millimeters. Therefore, in fact, photodynamic therapy is used for superficial tumors such as skin cancers. It is investigated also for brain tumors, for glioblastoma. Of course, delivering in-depth light is more invasive, because either it is performed intraoperatively with the open skull, or optic fibers has to be stereotactically inserted within the tumor in order to deliver the light into them. As a matter of fact, the progression-free survival is increased in these patients. So the concept is very interesting, of course, because the selectivity of the drug is very peculiar. But of course, in order to become less invasive, other forms of energy should be investigated, such as radiations, which are harmful. But in general, now sonodynamic therapy is picking up a lot of interest, with few trials ongoing. The concept of sonodynamic therapy is very similar. Basically, the idea is to deliver the energy using ultrasound, which have a higher penetration compared to light, in order to deliver energy depth in tissue, deep in tissues. There are many mechanisms of action that have been envisaged for the generation of reactive oxygen species in the induction of cell death, which are basically the concept of sonoluminescence, sonomechanics, and pyrolysis, which in turn induce different by-effects. Sonoluminescence, in particular, resides on the idea that microcavitation can generate photons deep in tissues, and the light generated deep in tissue can in turn activate the photosensitizer, the sonosensitizers. Also, the sonomechanics, the movement induced by ultrasound, it is thought to have a role into cell death. The generation of reactive oxygen species has been proved in different studies. This is a recent paper published in 2021, showing the sonodynamic therapy group in glioblastoma, human glioblastoma cells, showed a higher percentage of reactive oxygen species, an oxidized product within the samples. Hyperthermia, it was thought in the past to have a role, but recent studies show that it doesn't really play an important role into the effects of sonodynamic therapy. Cytotoxic effects have been also proved in different papers. This is a recent study conducted at the University of Virginia, published in 2020, showing how apoptosis and necrosis are induced in the sonodynamic therapy group compared to 5-LA or FUS alone in human malignant glioblastoma brain cells. The sonodynamic therapy also is thought to induce an anti-angiogenetic mechanism, with various studies supporting this idea. This is an example in a mouse model where they showed a reduced expression of a vascular endothelial growth factor in the sonodynamic therapy group compared to control 5-LA or ultrasound alone. And also a very interesting effect by effect is the effect of sonodynamic therapy on the tumor infiltrate, lymphocytic infiltrate, and on the tumor microenvironment, showing how the host T cells are activated against the tumor, and also the tumor suppression, myeloid suppressor cells are inhibited by sonodynamic therapy. This is one of the first, probably the first in vivo study conducted by Professor Umemura's group in Japan, showing the efficacy of sonodynamic therapy in vivo in an intracranial glioma model in rat, showing the destruction of the tumor, the regression of the tumor in time with sonodynamic therapy. And this was confirmed later on by Professor Kobayashi in Japan as well, who proved both in vitro and in vivo that sonodynamic therapy with 5LA was effective, and they used an MR-guided system to descend. Safety and efficacy, safety and feasibility of sonodynamic therapy using an MR-guided system was proven in this study conducted at the University of Virginia, sponsored by the Focus Ultrasound Foundation, where a large animal brain was sonicated in multiple locations after the administration of both 5LA and fluorescein, showing no damages to the healthy brain. This is the study that we are running in Europe. It's actually the only study in Europe about sonodynamic therapy. We are investigating the use of oral 5LA, performing treatment with an MR-guided system. And this is a safety and a feasibility study, and this is the only study among the others that is tackling newly diagnosed glioblastoma and not recurrent glioblastoma. As I mentioned, we're involving newly diagnosed glioma, not larger eventually than 5-6 centimeter. The patient received the drug 5 hours before starting every procedure, so that the peak is around 7-8 hours during the treatment. And we use a low frequency system to perform the sonication. And the patient is then followed for up to 3 weeks with MR, seriated MR and neurological evaluation, and then undergo surgical resection, and then follow up, of course, and we analyze the histopathology. And then the patient enters this not regularly the STOOP protocol. As I mentioned, the primary outcome is the safety, which is assessed by imaging and clinical evaluation and histology, and the feasibility, calculating the amount of treatment of the tumor that we are able to treat, and the secondary outcome is the efficacy. And as you can see, there is a variegate crew that take care of the patient and the treatment. This is the first case that we treated. It's a bit of it's a left frontal glioblastoma, not the ideal case probably to start with, but we had to start somewhere. The patient is prepared as in a regular focus ultrasound procedure with a stereotactic frame, and then it is placed in the MR bore. This is the planning of the treatment, and as you can see, we aimed at having a pretty tight grid in order to cover the whole area that we are sonicating, with an averaging a power of 40 watts per square centimeter. And each sonication lasts for at least 150 seconds, and it is repeated, in order to reach a sonication time of 16 minutes per spot. These are the overview. This is the overview of the first sonication, which lasted 3 hours and a half, and we treated 1.5 cc. So the first experience was a bit cumbersome. Then we improved our workflow. This is a 3D scheme showing the pre and post sonodynamic therapy with some of the targets, the main targets. And in this magnification, you can see how on the left we have the contrast SWI showing the tumor with some necrotic areas, and immediately, 24 hours immediately after the treatment, the necrotic areas were much larger, corresponding to the area which received the treatment. The patient underwent surgical removal of the tumor, of course, with ultrasound guidance, and you can see the bulk of the tumor here, and with neuronavigation in order to localize precisely and perform selective biopsy samples of the sonicated areas. And of course, we used 5LA as an intraoperative guidance. This is the postoperative CT and postoperative MRI showing the removal of the tumor. And on histology, we noticed how the treated area, which were analyzed separately, showed an increased amount of activated lymphocyte, and also reduction in myeloid suppressor cells environment. So in conclusion, there are many preclinical studies proving that sonodynamic therapy is effective and it's safe and feasible as well. And many trials are ongoing, and we need to assess not only the feasibility, but also the efficacy. And we are really curious to learn from the other speaker about how the trials are going. For sure, there are big question marks still about the mechanism of action, parameters, monitoring the treatment, and also the targeting, which is, I think, a very interesting point, how focused and how precise do we need to be. And also, how should we administer sonodynamic therapy? Before surgery, instead of surgery, after surgery, as a maintenance, repeat multiple treatments. So still many questions open. And also, in the future, there are new sensitizers and new targets to be investigated. And as you can see from this panel, this is a huge choral effort, and thank you for your attention.
So thank you to the National Brain Tumor Society and the Focus Ultrasound Foundation. Really is an honor to be part of the webinar, and we've had such great experience with these groups really pushing the field forward. Obviously, for us, ultrasound is the core of our technology, but these two groups, I think, have been at the forefront of just development in general and the fight against brain cancer. So in addition to running Alpheus, I also have an academic title. So I run the Brain Tumor Center at the Albert Einstein College of Medicine. So I run the Brain Tumor Center. I'm also the chief of the Skull-Based Center here in New York. And so kind of have a dual role with my foot in the academic world as well as in the innovation world, running Alpheus Medical. And really the story with brain cancer is the fact that it's just, we all know it's extremely hard to treat. One of the questions is, why is that the case? Why is it so hard to treat? And in my clinical practice, it's not uncommon for a patient to come in with enhancement on a scan, and then people from the in treatment in the community will really focus on these small areas of tumor. But as we know in the field, it's not this small area of enhancement that causes the brain tumor to recur. It's these larger areas of T2 flare and invasive disease. And this is why the survival for primary and recurrent brain cancer is so poor. And there's no approved therapies that really effectively treat the disease. And so the enhancing portion of the tumor is here. We take that out with surgery. We take that out with, we really target it with different treatment regimes, but it's really this flare, T2 flare signal around that causes the recurrence and ultimately the cell death. And it's easy to focus on just the small area of enhancement, but it's this very large area in T2 flare that ultimately makes this an extremely difficult disease to treat. You can even see it encroaches across midline structures, corpus callosum, et cetera. It's notorious for attacking both sides of the brain. And so this is really a diffuse disease. And it's the reason that the 5-year survival rates for brain cancer are abysmal. So we've made strides in almost every type of brain cancer, or every type of cancer that exists except brain cancer over the last 100 years. And the reason is because we focus on treating small areas of enhancement within the tumor. But this is a full brain, full hemispheric disease, and we have to treat it as such. This is an example of Harvey Cushing's 2000th brain surgery about 100 years ago. And what's interesting is obviously grainy, but the way that Harvey Cushing is taking out this tumor, very similar. I just finished a case. The techniques are the same techniques that we use even to today. And the goal of this video is to really show that we really have not made progress significantly in this field. I mean, if we're resecting brain tumors the same way Harvey Cushing was resecting brain tumors 100 years ago, we really need advancement in the field. And diffuse diseases require diffuse solution. And that's where we focus on low intensity diffuse ultrasound. You really have to target the entire hemisphere. You have to target the T2, the flare, and even the tumor that you can't see. There are papers from the 90s showing that even up to 4 centimeters away from the tumor bulk where you think there's no disease, if you culture those cells, you'll grow brain cancer. And so it is short-sighted to think that we could focus on a small part of the tumor and expect to extend survival. We really have to focus on full hemispheric and potentially full brain disease to really effectively target brain cancer. What we do is sonodynamic therapy. So we used an FDA-approved drug. So for those of us in the field of brain cancer, 5-ALA is it's taken orally. It's 20 milligrams per kilogram. And this drug is used for fluorescence. So it actually helps us identify brain cancer during surgery. So we use the same drug with a proprietary ultrasound that delivers this low intensity diffuse ultrasound. So it truly is a combination product with the drug and the device. And the way that we treat is the patient drinks the 5-ALA in the morning, just as they would for brain cancer. 5-ALA causes the protoporphyrin IX to accumulate in the brain tumor. The ultrasound activates the protoporphyrin IX, which is the breakdown product of 5-ALA that is selectively accumulated in the brain cancer, to undergo cell death. And that's really the core of our technology. We don't require any MRI imaging. It's done as an outpatient. There's no anesthesia, and it's diffuse treatment throughout the entire hemisphere, which is one of the reasons it really excites us. You know, diffuse diseases require diffuse solution, and that's one with which we provide. There are great talks here as part of the webinar about why 5-ALA preferentially accumulates in high-grade glioma, and it really is a multitude of mechanisms. And what's important to know is that a lot of the ways with which brain cancer evades treatment with chemotherapy those mechanisms are not maintained with sonodynamic therapy. And so, namely, sonodynamic therapy can target some of these shortcomings of potentially things like resistance. And this is also a very exciting field of sonodynamic therapy. One of the reasons I think it's been very well adopted and very readily adopted is it's similar to photodynamic therapy in some conserved mechanisms of action. And photodynamic therapy uses light instead of ultrasound to activate cell death. And that is approved for a variety of diseases where light is very readily deliverable. So diseases of the lung, the esophagus, and the skin. And so there are preserved mechanisms. And the ultimate goal of sono and photodynamic therapy is to excite these triplet states: tumor cell necrosis, apoptosis, inflammation, microvascular shutdown in brain cancer. And that's really preserved between photo and sonodynamic therapy. What's exciting as well is within photodynamic therapy, anecdotally, if you're able to deliver light into the brain through invasive means, Survival anecdotally has been very, very, very long, but the problem is delivering light into the brain is an invasive procedure and can really only target these focal areas of disease. And as I've said multiple times, diffuse diseases require diffuse solutions. And so we look at sonodynamic therapy as the ability to deliver a photodynamic type therapy through the entire hemisphere. And the literature and the basic science studies will show that there are preserved mechanisms between photo and sonodynamic therapy. And when you actually look at the emission spectrum of the sound, it overlaps with the absorption spectrum of the light. And so there's reason to have excitement about the preservation of the mechanisms. Ultrasound as well is very well accepted in the brain. As neurosurgeons and people in the field, we're very comfortable with using ultrasound in the brain, which is another reason why I think it's been well adopted. So sort of as a summary of the technology in general, SDT is triggered using a low intensity diffuse ultrasound. We have found in our work that you can activate SDT without microbubble cavitation, with or without, and it's really promising and has now entered clinical trials for multiple clinical trials for high-grade gliomas, which is an exciting field. But I think that this really highlights the difference between photo and sonodynamic therapy. Really addressing the shortcomings of delivering a broader field, whole hemispheric treatment in a non-invasive fashion. And so I think that that's really the biggest difference. But I think one of the things that we suffer from in the field is an inability to have good models for brain cancer. The best that we have found in our work is an ability to treat canines. They're the only mammals other than humans to get naturally occurring brain cancer. It actually is more deadly than human brain cancer, with a lot of preserved genetics, particularly in the pediatric populations in humans. But this is not a model. I hesitate to call it a model because it truly is a very regimented clinical trial. The histopath and the imaging, skull thickness, skull density, much of that is preserved with very, very poor survival. So this clinical trial allows technologies to be tested with outcomes much sooner than in humans because the lifespan is much shorter. So we do this with the assistance of Dr. Pluhar and Dr. Arnold, the University of Minnesota. We focus French bulldogs, which actually have been found to have the worst survival. So we started testing FUSF technology approximately 2 years ago at the University of Minnesota, and they have a very large range of experience looking at different methods in their clinical investigation unit in canines, from personalized cell vaccine to temozolomide to immune checkpoint inhibition. And what we did is we took our human protocol and then we treated the way that we would treat in the canines. There are obviously slight treatments because of the difference in some of the anatomy of the skull, but as close as can feasibly be done, we have emulated the human therapy in the canine therapy. And what we have found is a very profound histopathologic efficacy. So you actually show cell death within the tumor, and you actually show upregulation of the immune system, so potentiation of the immune system. That's translated to a durable imaging response, which is translated to a significantly increased overall survival. So the survival in the French bulldogs, which we had found to be less than 2 months in general, we have French bulldogs living well over a year with significantly higher survival, many without any signs of tumor recurrence. So we took that data primarily at the request of our scientific advisory boards. Roger Stoop is the chair of our scientific advisory board. Walter Stumer is probably the biggest name in 5A-ALA. David Reardon, who leads Dana-Farber's neuro-oncology group, and Matthias Senge, who's one of the best-known basic scientists in 5A-ALA. And the thought was we could go into humans early, but why don't we test this in a large animal and really optimize the therapy, optimize the patient flow, and then that would lead to a much quicker ultimate pathway. And that's what we did. We took a step back at the advice of this group. We got into canines. We showed a profound effect and then moved into humans. And we've done all the things that you would expect of a company of our stage. So we have orphan drug, we have fast track designation, almost a decade of patents. And really the goal is to work with the FDA to help get this technology into humans. And we have done that with our first in human study evaluating 5A-ALA with ultrasound therapy for recurrent high-grade glioma. The three centers are Northwell Health in Long Island, Washington University in St. Louis, and Dent Neurologic Institute in Buffalo, New York. And so we have finished our first in-human study. We are enrolling an expansion trial as well for recurrent brain cancer. But as I finish the talk here, as I come up on the last couple of minutes, I think it's really important to address one thing as I finish. Why have we failed as a field in brain cancer? What have been the reasons that we have failed? And what clinical data do we find meaningful or valuable from the first in human trials to initiate randomized control trials? I think we've all seen enough failed phase 3 trials. How do we get out of this cycle? What data is valuable to investors? We can't do this without the investing community. We can't do it without the KOLs and large academic institutions. What clinical data do we need to indicate a successful registration trial? We have not been able to bridge that gap. And I think the reasons that we have failed there are multiple reasons. I think there are too many incentives, financial and otherwise, to hurry to a phase 3. If you really dig deep into papers from studies that have failed over the last 15 years, there's a very deep selection bias and inappropriate selection of historical controls. And I think that really needs to be addressed, and we really need to clean up the data and the comparative data to have any sort of data that I think is actually meaningful. And we need to separate surgical from non-surgical, or if we do, we need to subcategorize them. So I think it's really important to address the way that we do our clinical trials and really compare apples to apples. And I think so far we've really been comparing apples to oranges, or not even anything in the fruit world. So I think we really have to be diligent and responsible with the way that we conduct our clinical trials, even at the expense of time and finances. And I think that that's the only way that we're going to move the field of brain cancer forward. And I'm proud to say that that's hopefully what we're contributing to with our efforts at Alpheus. So with that, again, it's a real honor. So thank you to NBTS and the Focus Ultrasound Foundation. And I'm happy to answer any questions.
Awesome. Thank you, Dr. Prada and Agarwal, for those great presentations, and for Dr. LeBlang as well. Kirk, I am going to let you take over as the moderator of this discussion. We've got about 17 minutes left in the webinar, and I saw a couple of questions come in through the Q&A, so thank you to those folks who have already submitted. Please continue to do so as questions arise. Thank you so much again.
Yeah, thanks, Lauren, and thanks to all the presentations and the presenters. Really, really outstanding overviews. So one thing I just want to point out right away is, for full disclosure, is that Alpheus Medical is a portfolio company of the mission-driven NBTS Brain Tumor Investment Fund. So just wanted to be transparent about that. So one thing, let's from the audience, from Greg, he's talking about recurrent ependymoma. Could this technology be applied to diseases, lower grade gliomas, upon recurrence? So maybe Dr. Prada, maybe you could give a shot at that.
Well, yeah, that's a great question, and we've been thinking about using different sonosensitizers for different brain tumors. We started, I guess, from what Suzanne showed everyone, started with a glioblastoma, because we know from our surgical experience that 5LA is a sensitizer, and it is uptaken by 5LA. So for sure, we will achieve a sort of an effect. The use of sonosensitizers, the uptake of sensitizers in ependymoma is variable. So we need to probably first find the proper drug for each tumor. But yeah, this is a very open and interesting question. And this is why, for example, we conducted some studies using fluorescent, because we know from our intraoperative experience that fluorescent is able to highlight different histotypes. It's less selective than 5LA, but might be an option in the future. And it is a drug that is already available on the market also.
Great, thank you very much. So, Dr. Agarwal, there's a lot of buzz about sonodynamic therapy now, but it's taken a while for it to get to this point. And so, what are the reasons for that?
Yeah, so thank you again, Francisco. Very nice to see you and the whole group. This is just a fantastic event, and it's amazing to see how far we've come. We're having a webinar for sonodynamic therapy with the National Brain Tumor Society and Focus Ultrasound. 10 years ago, never would have even been in the conversation. And that's the reason why there was no buzz. So when we started years and years and years ago, I got an email back from somebody. This was when it was kind of a back of a napkin sketch of what I thought the device should look like. And someone actually responded back, a well-known name in the regulatory world before sonodynamic therapy was known. And he accidentally CC'd me on the email to his group. And he said, either this guy is crazy or he's brilliant, but I think he's crazy. So he's probably right. But, the thought of it back then was so foreign. You take this drug, it's going to accumulate in brain cancer, and we're going to selectively kill brain cancer cells with ultrasound. I mean, it sounds kind of sci-fi, but, with the work Francisco's doing and the work that, all the groups that you mentioned are doing, it's becoming more in the mainstream of an understanding. And I think that that's why there's a lot of collaboration between the groups, because any success is a group success in terms of understanding the technology and not being viewed as crazy. That's one reason. I think it's more in just people's minds. I get contacted all the time from people asking about sonodynamic therapy. It'd be unheard of a decade ago. The second is technology has really advanced over the last 10 years. Just think how we were doing treating brain tumors 10 years ago. I mean, it's a completely different world. If you look at the publications from 2010 and 2005 to 2010, after Roger's famous paper, it's a completely different treatment field than it is now. So I think the field has evolved, and also just the general thinking of sonodynamic therapy and its real true potential have evolved as well.
No, thank you very much. Just going to try to move quickly because there's a lot of great questions coming in. But one thing I'm curious about, Dr. Prada, is what's the story about different formulations? So you have IV, you have oral for 5-ALA, but also with fluorescein. So what's the thinking there?
Well, I think regarding 5-ALA, in Europe, there is a long-standing history of using 5-ALA for image guidance during surgery. So for people who are not familiar with that, basically, this drug is uptaken by the tumor, and with the microscope during surgery, we can use a certain wavelength, and basically the tumor turns orange, whereas the rest of the brain doesn't show any fluorescence. So it was natural for us and easier for us in Europe to use the oral formulation, which is the commercially available formulation. The IV formulation is not currently used in Europe for image guidance. It has been developed in the US, and it has been tested now for sonodynamic therapy. I'm not aware whether this has been used for image guidance. So, I mean, because we used to say that seeing is believing, we tended to use the oral formulation in our institution, and also because we have a huge perform huge research on the use of fluorescent. We feel that this can be another viable sonosensitizers that can be used for other histotype. But still, as I mentioned, so we're still trying to figure out what is all the mechanism of action are. So with 5-ALA. So I think first we need to fully understand the full potential of 5-ALA, and then maybe move to other options.
Great,
Kurt,
thank
you. Can I hijack?
Yeah, sure. Go ahead.
I'm sorry. I would love to interject on. Francisco made a great point about ependymoma, and I would just wanted to say one thing. Well, two things. It's pretty brutal to hear yourself give a webinar. I mean, is my voice that high? Do I let? So thank you for that. I appreciate you putting Francisco and I through that. The second is we actually had a lot of great questions, by the way, amazing. But we had a lot of dogs who actually ended up being low grade. If you remember, we did like 5-ALA dosed. You suspect high grade glioma. and then you treat. We had a bunch of dogs with resultant low-grade glioma, and the difference in the imaging between humans and dogs is dogs generally don't get contrast enhancement. They get T2 flare. So, and if you look at it during surgery, you get these tumors that are just absolutely fluorescing like crazy. And so this is getting into the peripheral invasive zone. This is getting into the T2 flare. And we have seen all the things that we've seen in the high-grade glioma being histopath efficacy, durable imaging response, and extension of survival. We're seeing that in the low-grade dogs that we eliminated from our data, of course, but we saw, at least anecdotally, accumulation of the 5-ALA, histopathologic efficacy with biomarkers of cell death and potentiation of the immune system, durable to imaging response, and increase in survival. So it is for the ependymomas and the low grade. And to be completely honest, Nader has done some fantastic work looking at low grade and fluorescence. So I think there's a lot of excitement for SDT in low grade and peripheral invasive.
Yeah, great points both. So one of the things that that always was compelling to me for sonodynamic therapy was the potential. It's non-invasive, right? So obviously being able to have a therapeutic benefit and profound improvements in survival is a key goal, right? We all want that. We want longer survival and potential cures, but the quality of life is a huge important thing. And so one of the attendees kind of went there. So I know it's early days, but Susie and you mentioned some of the data coming out, and then Dr. Prada and Dr. Agarwal, any quick, any early, certainly, there's a lot of data in the dogs now, but what are we seeing in humans in terms of side effects and things? So maybe, Susie, I don't know if you have any, you mentioned some in your presentation. So I don't know if you have any further comments there.
Yeah, we're very encouraged by the reports of senescence in their patients with glioblastomas as well as DIPGs, but I will definitely defer to Dr. Prada and Dr. Agarwal for their clinical experience.
I'm happy to start. We've done, standard 3x3 clinical trial design, which is so outdated. I mean, we really need to update this, but this is all sort of what we have to do. No duration limiting toxicities. We haven't seen any sort of safety concerns to limit any sort of our treatments. Patients do extremely well with the drug and the combination therapy. And I think the reason for that is because the drug has decades of data. I mean, approved for over 10, 15 years in Europe, Francisco's backyard. It's just, there is so much data on the drug with selective accumulation in brain cancer. The ultrasound we know to be very safe. We're non-ablative ultrasound. We don't ablate, we just treat in a very safe sort of fashion. And if you look at there are studies that actually show that ultrasound alone will do things like raise the seizure threshold. And there are benefits to the ultrasound alone in terms of the quality of life. We've looked at biopsies in the dogs in normal brain. We've had the scans reviewed in humans and in dogs, and with no untoward off-target effects. And I think the reason for that is 5-ALA selectively accumulates in the mitochondria. So when you have generation of reactive oxygen species, the reaction is actually happening within the mitochondria, within the cell. So you actually don't get these bystander effects and these off-target effects that you get from things like chemotherapy. So I think that's a big reason for the extremely safe profile that we've been seeing in SDT.
Yeah, that's encouraging.
I can comment on that. I mean, we did some preclinical safety study with both 5-ALA and fluorescein, and we targeted the normal brain because, as Vijay pointed out, it's difficult to find the good glioma model in animals. So we as an large animal, we used pigs, so there was no tumor. So we sonicated the normal brain. We didn't elicit any, we didn't observe any problem whatsoever, and nor we found any sort of cavitation. And in our 2 patients that we treated, we just observed a necrosis in the sonicated area, but even targeting the viable enhancing portion on T1 gadolinium and SMR portion of the tumor, the patient did well, and they kept going until surgery 2 and 3 weeks after treatment. So the safety profile was very good. And also we have a huge amount of the literature data from photodynamic therapy, which is safe as well.
No, thank you, everybody. So, I mean, unfortunately, I have like 5 minutes left, but there's so many more questions that I'm getting, it's like combinations I'm really interested in. But one thing that came in from the audience is, so we talked about now lower grade ependymoma and astrocytoma things there, but what about metastatic disease, or even pushing it beyond that, leptomeningeal disease? I mean, is there any evidence for getting uptake into the so anyway, whoever wants to take that.
Vijay.
Sure. Yeah, no, I'm happy to. So I think the best way to treat recurrent disease is by never letting the tumor become recurrent. Because, we've had a lot of, Francesco and I both have had, majority of our careers is spent having very difficult conversations. Let's keep you alive until your kid graduates from high school in June. Let's, those sorts of conversations. And I think that the reason we have those is because when we treat these patients with recurrent disease, even if you don't see LMD, leptomeningeal disease, there is leptomeningeal disease. If you have recurrent disease, these cells are laying in wait to grow and eventually to cause death. And I do think that our best chance is when tumors are primary. The patients are doing well. As David Reardon said, the brains aren't as beat up from all of these treatments. So that's my first comment, is that I think once we are treating in the recurrent leptomeningeal setting, almost it's too late, unfortunately. But I do think that there is promise in sonodynamic therapy for a couple of reasons. How many treatments do you have that selectively are able to pick out brain from normal brain? It's almost virtually no treatments that are available that are able to do that. The drug is extremely intelligent. So I have very cautious optimism for leptomeningeal disease and being able to treat. But it is a difficult disease because if you're in the CSF, you're in the neurospinal axis. It means you're in the spinal cord, you're in the posterior fossa, you're supratentorial. So I think once you are leptomeningeal, there are very few things that I think can potentially help. And our best chance is to prevent the disease from becoming leptomeningeal in the first place. That's what I think.
If you don't mind me interjecting for a second, Dr. Prada is doing some important work with the newly diagnosed GBMs, and Dr. Agarwal and some others are doing the recurrent GBMs. And to defer to what you just said, Dr. Agarwal, could you foresee this becoming a biweekly or monthly treatment from the minute they're diagnosed before surgery to get the enhancing portions as well as the flare components taken care of, after surgery doing it, and then throughout the treatment? If you really want to prevent the recurrence, just start from the beginning and keep going.
Yeah, I couldn't have paid you for a better intro. So in the beginning of next year, we're starting a newly diagnosed GBM trial treating monthly. And the goal is the same treatment regime we're doing in the recurrent. And so we are getting all the things, the IRBs, et cetera, ready to start enrolling. We're hoping it's going to be in Q1 of next year, so pretty soon. And that's exactly the goal. Let's do Stoop protocol. And then let's, add to the standard of care in newly diagnosed, and let's treat them monthly. Let's turn this acute terminal illness to chronic manageable disease. Assume if we turn glioblastoma into hypertension or, diabetes, which are horrible diseases, but ones that I think we can effectively manage with treatment and therapy. And so I think that that's our best chance. And that's our next step, is to go into the newly diagnosed population as a maintenance treatment.
So can I ask you, so basically you're going to treat the patients after a section of the tumor, or and this is going to be an add-on to the Stoop protocol?
Yeah, the problem with recurrent is
it's not an upfront treatment. It's not going to be an upfront treatment.
Correct. It's going to be a maintenance treatment. And I think, the thing is with newly diagnosed, it's just we have a standard of care. Although, Stoop will be the first one to tell you that it's a poor standard of care. He actually said at this meeting we had in January, he said, someone's got to do better than me, being him. But there is, we have to follow the one thing that has given us weeks of extension of survival. So it'll be an add-on to the Stoop protocol.
Okay. Now, because following quick question about the non-invasiveness. I think that, and what we see from our results, I think that this can be, sonodynamic therapy could become also an upfront treatment.
Absolutely.
Because but the problem is with our study is that we are not delivering enough treatment to the patients because we have there are technical constraints, of course, but maybe with a solution that can that is more easy to use or a wearable device, this can become an upfront therapy, and people eventually will not get surgery.
So that's, my dream, Francesco, my dream is to put you and I out of business, honestly. Like, I would love everybody to know we could do shunts or something. Yeah, no, we could focus on something else, spine surgery maybe. But I think, if we could eliminate the need for surgery and radiation, that's my dream. And this has nothing to do with the company or nothing to do with what we're doing. This is my personal, what do people put on Twitter? Views are my own, tweets or X's are my own, something. So these are my own. I would love to be unemployed from a neurosurgery standpoint. I really would. That would be my dream. And then to replace radiation completely. That is my personal absolute dream. And I think being in an upfront setting is so smart. But that's my ultimate goal. But I think we have to do it in a bit of a graded fashion and be responsible to sort of the regulatory aspects. But Francesco,
I think Vijay, I think there's ways to do that, right? One of the things that we have in BTS is this research roundtable program where we work with the FDA to talk about barriers for new product development in brain tumors. And so one of the things is, how do you move into the upfront setting in an ethical sort of way? That you could potentially do maybe you could do sonodynamic therapy to see and then look for responses with people, so I think there are creative ways to do this. It's just, as you mentioned, it is going to be stepwise, but I think we have to start thinking about how we get to that upfront setting and also being able to eliminate radiation, right? I mean, I think that's another big key there. So I know that we are over time. I want to be respectful for everybody's time. I also want to just say to all of the audience participants, I'm sure that the Focused Ultrasound Foundation, National Brain Tumor Society, Dr. Agarwal, Dr. Prada would be willing to go, respond to any of your questions via email or however mechanism that you do it. I'd also like to really, again, thank the Focused Ultrasound Foundation for having this webinar and having NBTS to be a partner in this. And, again, we should have probably had a 2-hour webinar because I think we could have these questions going back and forth for a long time. And perhaps we'll do that in coming up. But I'll turn it back over to Lauren now because, again, I want to be respectful of everybody's time.
Thank you. And I would like to thank the National Brain Tumor Society and you, Kirk, for being here and for supporting this webinar as well. So very, very appreciative of that. And what a great discussion. The time just flew by. Thank you all for being here. Thank you for sticking around if you're still here 4 minutes over time. But this concludes today's event. If your question was not answered, as Kirk mentioned, we will try to reach out to you to get those answered. And of course, if not, please just reach out to us. You can email us at info@fusfoundation.org. And stay tuned to our newsletter and website for invitations for future webinars and online events. Thanks so much for being here.